Cells communicate with each other and their environments in a variety of ways. One of these ways is through cellular adhesion molecules called integrins, which are present on the cell membrane. When an integrin comes in contact with special molecules called ligands on other cells or surfaces, the two form a bond. Integrin interactions are weak, so other integrins move along the membrane to the area of initial attachment to form more bonds and create a stronger adhesion. When interactions like this happen, chemical pathways within the cell that control actions such as cell growth or division are activated. But how exactly are integrins and other cellular adhesion molecules transported along the cell membrane? The answer lies in structures called lipid domains. Cell membranes can be made up of two or more different kinds of lipids, each of a different thickness. The difference in thicknesses can cause the hydrophobic tails of the lipids to be exposed to a hydrophilic environment. This leads to association of different lipids and to island-like formations known as lipid domains, or sometimes referred to as lipid rafts. It is believed that cellular adhesion molecules and other proteins are often attached to lipid domains. These adhesion molecule lipid domain complexes can travel along the cell membrane. The formation of lipid domains depends on interfacial energy. Interfacial energy is the amount of energy needed on the surface of a liquid for that liquid to keep its shape. A low interfacial energy results in less lipid domain formation, while a greater interfacial energy results in more lipid domain formation. Researchers in the Mechanobiology Laboratory at Penn State studied techniques that could be used to manipulate the formation of lipid domains. Researchers ran computer simulations using lipids called DPPC and DUPC. The researchers investigated how compounds called Amphiphiles would affect how much the DPPC and DUPC would form lipid domains. The three Amphiphiles tested were vitamin E, a compound called Triton X, and benzyl alcohol. Researchers then tested artificial membranes made of DPPC and another lipid called DOPC. The results showed that vitamin E lowered the mismatch between the thicknesses of DPPC and DOPC, and thus lowered the interfacial energy. This means that the presence of vitamin E in a cell membrane makes lipid domains less likely to form. Benzyl alcohol, on the other hand, increased the mismatch between the thicknesses of the lipids and effectively increased the interfacial energy. This resulted in more lipid domain formation. Triton X was observed to have a similar effect to benzyl alcohol, although the impact was not quite as noticeable in comparison. By using these three compounds, scientists hope to be able to control the formation of lipid domains in the future. Benzyl alcohol and Triton X can promote the formation of large lipid domains and effectively bring together adhesion molecules. This would greatly increase the chance of the formation of a cellular adhesion. Conversely, vitamin E can prevent lipid domains from forming large groups and decrease the likelihood of an adhesion forming. This new knowledge of how to manipulate cellular adhesion through lipid domains will allow researchers to perform new experiments to further understand the role of the cell membrane and may someday aid in treating disease.